I'd like to welcome you all to Amy's defense today, and she has a committee of 6, and half of her committee are her advisors. She has 3 co-advisors. I am going to be the briefest and introduce the other real co-advisors. So Amy's thesis involves pilot whales, genetics, and acoustics. And you'll see her bring these things together in a wonderful way. I have 0 expertise in any of those topics. So in this case, Amy has been my instructor over the last 5+ years, and I just want to thank you very much for teaching me so much about those topics and doing such an interesting thesis. So I'm going to introduce the real advisors. First Jay Barlow, and then Phil Marine.
Thank you, Lisa. And it's a great honor to be here and introducing Amy Benzies, who's done, as Lisa said, a fantastic job with the dissertation, but you'll have a chance to judge that for yourself. Amy came to us from a diverse background. She got her undergraduate degree at University of New Hampshire, and it was a dual major in journalism and biology. So all I have to say to people who are looking to take on students in the future at some point in their career, if you get a chance to take on a journalism major who's actually worked in the field as a reporter and had to turn out copy on a daily basis, I'd say yes to that student, because they've learned the discipline of writing. You don't have to teach them grammar, and they in fact already know how to write, which is a great blessing. After that, she went to Peru and did 2 years in the Peace Corps. Also, if you get a chance to hire someone who's been in the Peace Corps, it teaches them hard work under adversity, and I strongly recommend the traits that they learn there. Then she came to the Southwest Fisheries Science Center and worked for a number of years in our Antarctic division. It was more or less an administrative assistant position, but she managed to turn it into a position where she was logistic coordinator for all of the Antarctic research that was done by that division. So she was handled the ships and shipping and all that sort of stuff. That wasn't enough work. During that time, she volunteered in Phil's genetics lab, and she learned genetics in my lab on a volunteer basis. And I think she had in her mind that she would actually get to decide which of those 2 things she liked better, and maybe she'll tell us later on today. But so far, I haven't seen any sign that she's actually made up her mind yet. The next person I'll introduce will be Phil Morin, her genetics co-advisor to this dissertation project.
Of course she likes genetics better. It's a pleasure to be here. And Jay said it, having someone come in with the tremendous diversity of background that Amy had coming in was really nice, because she already had some great ideas of what she wanted to do. She was wide open to all sorts of different opportunities and different suggestions, but also had the experience to be able to say, I can do that, because she'd done so many things before. If there was one word I could choose to describe Amy, I would say adventure. She clearly loves to try things out and does not shy away from new experiences. So my part of this, I get to show a few pictures. But before I do that, I want to let you know about the logistics of today's program. Amy's going to give her dissertation defense talk. Is this going in and out, or is it just my voice? And right after that, you will have an opportunity to ask her questions. And so we'll have an open question and answer period, and then we'll ask everybody to leave the room except for her committee. So her committee can talk to her in private and ask questions. And then following that, we will all meet down at Surfside for a celebration. So Amy Van Sythe, this is your life. No, just kidding. Amy, your mouse doesn't like me. It's not. I can't find out. Oh, okay. Thank you. This is how it always goes with students and advisors. So Amy has had a chance to learn a lot of skills. So working in the field, she clearly has embraced different challenges. I don't have any pictures of a genetics or acoustics lab here because they're really boring places to take pictures. So, but Amy is not afraid of trying out new things. So everything here from running big machinery and ships to woodworking. She also is not afraid of living in pretty extreme environments. She's lived in Antarctica and the Arctic doing field work, as well as some warmer places. And her taste in clothing is usually pretty good, but I'm not sure about the one on the bottom right. She also knows how to have a good time, apparently. This is what conferences, after all, are all about. She's made the most of her experiences out on the ocean, doing a lot of different types of research, as well as on land, doing things that are both exciting and scary. And today she's going to tell you about the adventure of the last 6 years, where she's been working with pilot whales. And with that, I'm going to turn it over to Amy to talk about genetics and acoustics of pilot whales.
Can you guys all hear me? Can you hear me now? Yes. Okay, good, because I hate this one. All right. This guy's really good.
Amy, you have to speak up.
I'm going to speak up. Thank you guys all for coming. It's really nice to see so many friendly faces in the audience today. And I'm really excited to talk to you about what I've been doing for the last 6 years, which is studying gene-culture coevolution in a social citation. But before I get started, I want to tell you all a secret. That secret is that the first rule of loving whales is that you don't talk about loving whales. So today we're going to talk about acoustics and genetics, population structure. We're definitely not going to talk about this adorable guy right here, the short-fin pilot whale. By the way, it's not a whale. Short-fin pilot whales are actually in the dolphin family. They're the second largest member of the dolphin family, second only to killer whales. And they look kind of like killer whales as well, except that they're all black. But unlike killer whales, we don't know very much about short-fin pilot whales because research funding is extremely limited. So, although we don't know very much about these animals, they face a wide variety of local and regional threats. And in order to manage those threats, we need to know a lot more about their ecology, their evolution, and their behavior. And one thing that we do know is that they are found throughout the world's oceans in both the tropical and temperate areas of the ocean. We also think that they eat primarily squid, and they dive up to 800 meters to hunt for their food. Now, these animals live and hunt in groups of 15 to 25 animals, and based on photos, we know that they're nearly constant companions for periods of a decade or more. And as they're hunting, about half of the group will break off from the surface and dive down to depth. And while they're hunting, they will continuously make social calls between them, so that when the group that's hunting comes back to the surface, they can find each other in space. And it's social behaviors like this that may affect the vocal repertoire of these animals, and may even go so far as to affect their DNA. So describing the genetic and acoustic patterns that we see within the species is going to give us a better understanding of what drivers may be affecting their population structure. So today I'm going to start with an introduction to what population structure is, and then talk a little bit about the field methods that I use. In the first section, I'll cover geographic structure in the Pacific Ocean, and that section comprises 2 of the chapters in my thesis, one on acoustics and the other on genetics. And then in the second section, I'll focus in on the Hawaiian Islands and talk about population structure there. And again, this covers 2 chapters in my thesis, one on acoustics and the other on genetics. And then to wrap up, I'll give a little summary of the results and talk about why it's important. So I just want to take a moment now to mention that of the 4 chapters I'm talking about in this thesis, the first 2 have been published. The third one was recently submitted to molecular ecology, and the 4th is in its final stages of preparation and will be submitted in the next month or so. And I had the opportunity to work with some really great co-authors on all of these chapters. My primary co-authors were my advisors, Bill Morin and Jay Barlow. I also worked with Robin Baird and Seda Mahaffey from Cascadia Research Collective, who provided a lot of the data and helped me with a substantial portion of the analyses. And Karen Martin from NOAA and Marie Roche from Scripps were both instrumental in helping me analyze the patterns that I found in the acoustic and genetic data. So if I say we during the presentation, these are the co-authors that I'm talking about. All right, so let's dive right in. And I'll start by telling you a little bit about population structure. So I think the idea of population structure may have first been introduced by Charles Darwin, who said that those forms which are so closely similar to some other forms are in several aspects the most important forms. We have every reason to believe that many of these have permanently retained their characters, for as far as we know, as have good and true species. And here what Darwin is talking about is population structure and why it's important to species evolution. So for the purposes of our discussion today, I'm going to define population structure as systematic differences in characters among groups within a species. Now, depending on what character you choose to examine, you're going to see different differences in the resulting population structure. So in the example here, if I choose character or shape as the primary character of interest, then I would find 2 populations. And if instead I chose to consider color, I would find 3 populations. And then finally, if I looked instead at the geographic location of the individuals, I would find 4 populations. So today I'm going to talk about acoustics and genetics as 2 characters you can use to examine population structure. And while genetic structure evolves at a rate of multiple generations, acoustic structure may change much faster, as it's learned during the lifetime of an individual. So comparing these 2 types of data is going to give us important information about longer versus shorter term population structure, each of which has important management implications. So let's start by looking at how genetic structure arises. So let's say that this phrase here represents a portion of your genetic code, which we're going to call a sequence. And then each character in that sequence is called a nucleotide. And we'll give that sequence a label, and that label is called a haplotype. Now, if a mutation arises in that sequence, we'll call that mutation a single nucleotide polymorphism. And we'll give that sequence a new haplotype label. Now, over thousands of years, mutations will start to build up in the genetic structure. Each new sequence will be given a new haplotype label. So within a population of individuals, all of the haplotypes should be distributed randomly. because there are no barriers to mixing or mating. But if a barrier forms, or say a small group breaks off and moves to the other side of the barrier, then those two groups can't mix anymore. And over time, their populations start to look different through a process called random drift. The frequency of haplotypes will change on each side of the barrier, independently of what is happening on the other side. New haplotypes may arise, and others will be lost. And without any mixing, those haplotypes will remain separate from each other. So that over thousands of years, what started as one population becomes two. Okay, so now I'm going to talk to you about the same process for acoustic data. But before I do that, I wanted to show everybody a spectrogram so you can get an idea of what sound looks like. So this spectrogram shows time on the x-axis and frequency or pitch on the y-axis. If you follow along with this red bar here, I'm going to play the sound for you and you can see what it looks like. Turn the volume up a little bit. Play it one more time. All right. So one way to think about acoustic structure is to think about the many different call types that an animal can make. So this is an example of one call type. Within a population, there can be hundreds of different call types. And call types can be lost, and new ones can arise over time. We can also think about the shape of each of the calls and how that might change over time. And that change is going to happen through a very similar process as the one we saw for genetic structure. So a barrier forms, and random drift on either side of that barrier causes the sounds to change in shape. For example, they might change in duration, or you might see a change in frequency. We can measure things like start and end frequency, number of inflection points, slope, and duration of the calls, and look for systematic differences in those characters to detect population structure among groups. Now, one important difference from in acoustic data from genetic data is that with acoustic structure, you're talking about something that's going to change much faster, because this is the vocal repertoire of an animal is learned during the lifetime of an individual, either from the parent to the offspring or socially from other members of the group. So that change is going to happen on the scale of years or decades rather than generations. One important commonality, though, between these two types of data is that the structure we see arises because of some sort of barrier. And that's what I'm going to focus a lot of my time on today. Because understanding the population structure of a species means understanding the geographic and ecological barriers that cause that structure. And oftentimes, population structure is caused by geographic or ecological barriers, like distance, current, bathymetry, temperature, or prey distribution. And these kinds of barriers are stable and are stable enough for long periods of time that through genetic divergence and speciation, new population structure can emerge. New populations and then species can evolve. So the diagram on the left here shows a good example of a terrestrial barrier, where a river forms a canyon through what was once a continuous forest. And on either side of the river, the trees in the forest begin to diverge into new populations and eventually into species. But compared to land, there aren't a lot of geographic or ecological barriers in the ocean. That's especially true for large predators like short-finned pilot whales, which can cover huge distances. And yet, sometimes we still find evidence of population structure. So we have to ask ourselves, in the absence of physical barriers, why do we see population structure? And to answer that question, I relied on the principles of gene-culture coevolution theory. This theory essentially states that genes and culture can be so strongly linked to each other that they each influence the evolution of the other. Now, this can happen in a broad sense when a cultural or social behavior separates two groups so that they don't mate with each other. Or it can happen in a narrow sense when a specific gene is tied to a specific behavior. The great example of this is lactose tolerance in humans, which evolved with the advent of dairy farming. So traditionally, gene culture coevolutionary theory has been reserved only to humans because we thought that animals didn't have social or cultural behaviors. But that's been proven wrong for an increasing number of species. And in fact, we see socially driven fine-scale acoustic structure in a variety of social mammals, including primates, elephants, rock wallabies, prairie dogs, killer whales, and sperm whales. Killer whales are probably one of the most well-known examples of this phenomenon in the marine realm. In this species, several lineages have radiated out in the last 250,000 years. That radiation has been primarily driven by social behaviors like hunting strategies and vocal repertoire. We see a similar pattern in sperm whales, where multi-level societies have emerged, and that has also primarily been through cultural transmission, this time primarily the vocal repertoire. And those societies have been stable for long enough to cause genetic divergence within the species. Now, just as we've seen evidence that social structure can affect genetic structure, you can also see the reverse, which is that genetic structure can have a driving effect on the social structure of these animals. And that's through loyalty to family, or what we call philopatry. In this case, what we're talking about is that animals do not disperse throughout their range, as you would expect, but instead tend to remain with their family units for their entire lives. In matrilineal societies, ecological knowledge and leadership of the eldest female is likely a strong incentive for younger individuals to stay with their families for their entire lives. And in that way, the family becomes the basic unit of group structure in social species. Okay, so now that we've covered the basics of gene culture coevolution theory, I'm going to summarize by laying out the different links that I tested for in order to describe population structure in short from pilot whales. So we'll start with those barriers. Those prevent mixing among populations and give rise to genetic and acoustic structure, either through restricted mate selection or restricted learning of the vocal repertoire. Now, acoustic structure can affect genetic structure if an individual prefers to mate with someone that sounds the same as it does. And genetic structure can affect acoustic structure if individuals are more likely to pick up their vocal repertoire from their immediate family. Now, for geographic and environmental variables, we can stop right there, because these types of barriers are not usually changed by the population interacting with them. But on the other hand, barriers created by social behaviors can change as the population changes. But if genetic structure and acoustic structure work to reinforce the current social structure through things like family philopatry and group identity maintenance, then social structure can become a barrier that is stable enough to change population structure. So these are the links that I tested for when I examined population structure in the Pacific Ocean and in the Hawaiian Islands. So throughout the talk, I'll refer back to the slide to help orient us all as I test the specific links. All right, so to collect the genetic and acoustic data that I used for my thesis, I joined several different field projects in the Pacific, in places like the California Current, the Gulf of California, and Hawaii. And as is the case with many marine mammal field projects, we weren't focused just on pilot whales. Instead, we surveyed, counted, and sampled a variety of whales and dolphins. And to give ourselves the best shot at understanding the lives of these animals, which can only be observed for brief moments while they're at the surface, we use a variety of sampling methods. And the most common method is visual observations, but we also use photographs, biopsy samples, satellite tags, and acoustic recordings. Today I'm going to talk about just two of those, and I'll start with genetic data collection. So to collect genetic data, since we can't approach the animals close enough to take a skin sample, we instead use a crossbow with a modified arrow. The arrows that we have use a modified tip that takes a small skin sample. It's a little bit smaller than the first knuckle of your pinky finger. And the yellow float bounces off the body of the animal and helps the sample stay out of the water until we can retrieve it. Now, back on land, we divide the samples to be sent to several different institutions. About half of each sample is sent here to the Southwest Fisheries Science Center, where it's stored in a national archive at -8 degrees Celsius. To begin any project, samples are checked out from the archive and DNA is extracted. To extract the DNA, we first use an enzyme to break open the skin cells, then remove the cell debris and clean the remaining DNA with ethanol. A 2 mm piece of skin is enough to generates enough DNA for almost any sequencing project. So we can use every sample we collect for multiple different projects. All right, so now that we've gotten all of our biopsy samples ready for sequencing, let's move on to acoustic data collection. And there are a variety of different types of recorders available out there for collecting acoustic data. But the types that I use fall into two basic categories. The first is towed arrays, which are deployed from the back of the ship and then dragged along as the ship moves forward. And the second type are surface-mounted buoy recorders, which again are deployed from the back of the ship, but then remain in one place as the ship moves away. And these are usually placed in the path of the animals and left to record sounds as the animals pass by. And once we have the recordings, we take them back to the lab and visualize them on spectrograms. We then extract individual calls and classify them. And then finally, we trace each call so that we have a profile of the call contour that we can then use to measure features like duration, frequency, range, and slope. Okay, so we now have our data. So let's first use it to look at population structure in the Pacific Ocean. And just to refresh your memory, here are the components of population structure that we'll be looking at. So first we'll determine if there's any acoustic or genetic structure in the Pacific Ocean. And then we'll see if that's correlated with geographic or environmental barriers. And we can also look for correlations between patterns in genetic and acoustic structure, which may indicate links between the two. So the story in the Pacific starts in the 1980s in Japan, when morphologists described two different morphological types of short-fin pilot whale. These types had separate habitats in the north and the south of Japan, so they called them the northern and southern types. But after I did a little bit of digging, I found that these two types had actually originally been described in 1760 by a scientist named Yamase, and he called them the Shiho and the Niza types. Now, I just want to, I'm going to use these names throughout the rest of the talk. I just want to point out for some of you, because I'm sure you'll notice, that the first letters of these words is switched. So try not to let that confuse you. It confused me for about 3 years. So the primary difference between the Niza and Shiho types is the shape of their melon, or forehead area. You can see that in the Shiho types, they have a pronounced round melon, and Niza types have a very square melon. They're also different in body size, coloration, skull morphology, and number of teeth. Now, in 2009, a scientist named Marco Ramos and his colleagues used tissue samples from the two different types to examine their genetic relationships and found that they were genetically distinct as well as being morphologically distinct. And as I mentioned, the two types have a north-south distribution off the coast of Japan. That distribution is separated by the Kuroshio-Owashio extension current. The Shiho type being found in the colder waters of the Owashio current, and the Niza type found in the warmer waters of the Kuroshio current. So based on that evidence, scientists hypothesized that these two types might actually be distributed throughout the rest of the Pacific Ocean, with sea surface temperature driving their distribution. So they expected to find Shiho types in the colder water regions of the Pacific, and Niza types in more tropical regions. So, using the results of all these studies, I designed a study to examine the population structure of short-fin pilot whales throughout the Pacific. And I wanted to answer two basic questions. The first was whether there are genetically or acoustically distinct populations in the Pacific Ocean. And then, if so, how these populations relate to Shiho and Niza types found in Japan. So to do this, I collected mitochondrial control region sequences from 331 samples stored at Southwest Fisheries Science Center, augmented that with 155 sequences that I downloaded from GenBank, which is an online repository of DNA sequences. I also collected 12 acoustic recordings from the Eastern and Central Pacific and 23 from Hawaii. I began by testing for geographic structure among 6 seemingly isolated regions throughout the Pacific Ocean. That's these right here. What I found instead was evidence for 3 broad populations in the Eastern, Central, and Western Pacific. Now I had only a few, I had only a few samples from the Indian Ocean, but those also grouped with the Western Pacific population. And next, I wanted to look at how all the haplotypes I found were related to each other, so I built what's called a haplotype network. Now this figure seems a little complicated at first, so I'm going to walk through the whole thing with you. Each circle here represents a single haplotype, and the size of that circle represents the number of individuals that have that haplotype. The lines show which haplotypes are connected, and the red numbers tell you how many nucleotides are different between each haplotype. So we can identify the haplotypes that have been sequenced from pilot whales with NISA or SHIHO type morphologies and use that to get a sense of the distribution of the 2 types throughout the Pacific. So there are 2 known SHIHO haplotypes, and that's 5 and E here. And what I want you to notice is that in addition to being found in the North Pacific, those animals are predominantly found in the Eastern Tropical Pacific and the Eastern North Pacific. And then there are 4 known NISA haplotypes, and those are found in South Japan, Hawaii, Southeast Asia, the South Pacific Islands, and the Indian Ocean. So I used the haplotypes from known NISA and SHIHO pilot whales to map the distribution of the 2 types throughout the Pacific. I found that the 2 types had a distinctly east-west distribution, rather than a north-south distribution or one defined by sea surface temperature. I did go on to test the relationship between haplotype and sea surface temperature. I did that using a variety of regression models and ANOVA analyses, and I found no link between the 2. So it's possible that instead, these 2 types are separated by the wide expanse of the central North Pacific gyre, which is known to be a low productivity area. And in fact, it's such a poor source of food that it's often referred to as a desert. Another possibility is that their distribution follows the distribution of their favorite prey types. Now, like I said before, we actually don't know very much about what pilot whales eat, so this hypothesis is difficult to test at the moment. But we do know that some squid species have distributional patterns that match that of NISA and SHIHO pilot whales. So here's one example. And what you see here is that Humboldt squid, or Dosidicus gigas, has a distribution that matches that of the SHIHO pilot whales in the Eastern Pacific. And then on the left, there's a closely related squid species, Stenoteuthis, that has a distribution matching NISA-type pilot whales throughout the Western and Central Pacific. Now, this isn't a scientific analysis. I literally am just visually comparing the distributions of these 2 sets of species, or 2 sets of animals. But I think this hypothesis is worth researching further. Okay, so shifting to think about vocal repertoires, I can use what I learned from the genetic data to inform my experimental design for my acoustic data. So I divided my samples geographically to look for differences between NISA and SHIHO vocalizations. And first, looking just generally at their vocal repertoire in terms of the number and types of call types, I found a greater diversity of call types in NISA pilot whales than in SHIHO pilot whales. So NISA pilot whales also had a greater number of multi-component calls and more endemic calls. So the graph here shows a standard test for species richness, but here I've replaced species with call types. What you can see is that as sample size increases on the x-axis, the expected number of call types on the y-axis increases dramatically for NISA-type animals over SHIHO-type animals. On the other hand, SHIHO pilot whales had more non-tonal calls, which were buzzes and creaks without any defined shape. I then looked at specific acoustic features in the calls of NISA and SHIHO pilot whales and found significant differences in start frequency, range, and duration of calls. And I used those differences to build a mixture model classification algorithm. The figure here is an example of the model output using frequency range on the x-axis and duration on the y-axis. And in the left panel, what you see is the output of the training algorithm, And the circles here are the primary mixture for each of the two types. And then in the panel on the left, you see the output of the classification algorithm, which shows correct NYSE classifications in blue, Shiho classifications in red, and then incorrect classifications in black. Now, the model was able to classify calls to a specific type with 74% accuracy. And that not only indicates the strength of the differentiation between the two types, but it's also a potentially useful tool for defining distributional boundaries between these two types in areas where we don't have genetic or morphological data. And a great example of that is the Central Pacific Ocean. So to sum up this section, I found evidence for 3 regional populations in the Pacific Ocean: an eastern, central, and western population. And then I found that those 2 types described off Japan were distributed throughout the Pacific, with the Shiho type in the eastern Pacific and the NYSE type in the western and central Pacific. And these 2 are likely just separated by some sort of geographic barrier, like the North Pacific gyre, or the distribution of their preferred prey. I also found strong evidence for differentiation in their vocal repertoires, which likely indicates lack of communication and vocal learning between the 2 types, and may be used to identify distributional boundaries. All right, so now let's focus in on the Hawaiian Islands and look at population structure and its drivers on a local level. Coming back to this framework slide once again, I'm going to reorient you to what kinds of links we're looking at in this section. So, same as in the last section, we'll look for acoustic and genetic structure, and then look for correlations that would indicate what types of barriers are causing that structure. And in this section, I'm really going to focus on that social structure that I mentioned in the introduction through gene-culture coevolution. So for that purpose, I'm going to look for reciprocal effects of genetic and acoustic structure on the social structure. Those effects could stabilize social structure for long enough to accelerate divergence in population structure at a local level. So in Hawaii, short-fin pilot whales have been observed since about 2003. And the social structure of these animals has been described using photographs collected between 2003 and 2015. So based on this data, individuals, which are shown here in blue, were grouped into social units, which are in red, and then the social units were grouped into clusters. Now, clusters tend to prefer one island or group of islands, and they're all sympatric in that region. So because they all use the same habitat, we call these groups island communities. And there are 3 hypothesized island communities. One around Hawaii Island, one that extends from Kauai to Oahu, and then a third that extends from Oahu to Maui. So in this section, I want to answer the overall questions of whether there are links between genetic, social, and acoustic structure in short-fin pilot whales. And to do that, I'm going to answer 3 specific questions. First is whether social units and clusters are comprised of family members or randomly sorted individuals. And then in turn, whether we see evidence of socially or geographically driven population structure. And then finally, does acoustic structure reflect genetic structure? So to answer these questions, I used 254 skin samples, again from the Southwest Fisheries Science Center. I sequenced the mitochondrial control region from 242 of those samples, and then sequenced nuclear DNA from 112 of those samples. To sequence the nuclear DNA, I used what's called a capture array to target 78 loci throughout the nuclear genome. And using that method, I successfully generated 119 single nucleotide polymorphisms at 49 loci. I calculated population structure using standard FST indices, then estimated relatedness using a dyadic maximum likelihood estimator, and finally tested for a social genetic link using Mantel tests and linear regression. I additionally collected vocalizations from 22 recordings from throughout the Hawaiian Islands. But of these 22 recordings, 12 of them were with multiple groups, and so those had to be discarded. The remaining 10 encounters, the remaining 10 encounters represented 6 clusters found throughout the Hawaiian Islands. So to answer the first of the 3 specific questions in this session, I found that genetic relatedness affected social structure on multiple hierarchical levels. The graphs in this figure show our relatedness histograms, and what they show is you see distributions in each graph, and those are the distributions of expected relatedness values if these groups were sorted randomly. And then the red line in each graph represents the actual relatedness calculated for each group. What you see is that these values are all significantly higher than expected, and that indicates that these groups are made of closely related individuals. We interpret this to mean that social units, which are the smallest grouping of individuals, the social units represent closely related nuclear families. And then clusters represent extended family groupings. So I then tested for geographic stratification as a driver of population structure. And at the broadest geographic level, I found evidence for an insular population around the main Hawaiian Islands. And that population was separate from both the northwestern Hawaiian Islands and the pelagic regions surrounding the archipelago. Now within that insular population, I could only test for differences between 2 of the 3 island communities, but I did find differentiation between both of them, or between the 2. I then wanted to look at whether social structure affects genetic and acoustic structure. So I first examined genetic divergence between clusters, which, if you remember, are the purple circles here. Now I wanted to remove any signal, any potential signal from relatedness before I did this, since we already know that clusters are highly related. So I removed highly related individuals from the clusters, and then conducted 10 pairwise tests of differentiation between different clusters. Of those 10 tests, 8 were found to be significantly different, and a global FST also showed significant differentiation. Now, I did a similar test to look for socially driven acoustic structure. What I did was I used a multivariate analysis of variance to test for differentiation among clusters in start frequency, frequency range, and duration. And I also found a strong signal for differentiation. So we interpret this to mean that communication and mating or dispersal is restricted by cluster association, even though these clusters are sympatric in their distribution, so that within island communities, social structure is causing genetic structure. So all of the tests that I've shown you so far have been somewhat indirect tests. I look for patterns that you would expect to see if geography or social structure were acting as barriers in creating population structure. And then I look for evidence that genetic structure helps stabilize social structure. The patterns I've found support the idea that all 3 of these aspects of population structure are strongly linked in pilot whales. But I wanted to take this a step further and directly test some of these relationships. About one of those relationships, and that's the relationship between how related 2 individuals are and how likely they are to spend time together. So first here, this figure shows the results of a Mantel test of correlation between those variables. And you see the distribution of random variables on the left, or random values on the left. And way off on the right there, the red line shows the actual calculated correlation coefficient between relatedness and association. Now this was highly significant, and it indicated a strong correlation between how closely related the 2 individuals are and how much time they spend together. So the next thing to do was to test whether this link was causal. And to do that, I used the linear regression. I tested whether relatedness, distance, or a combination of the 2 affected the amount of time that 2 animals spent together. So this equation has a term for each of those and an intercept. Now, you have to remember here that my variables do not represent individuals, but instead represent relationships between individuals. So none of these relationships are totally independent of each other. So I'll just give you an example here. I have a relationship with each of my committee members, but none of those relationships is completely independent because I'm involved with every single one of them. So I need to control for the partial dependence of all of my variables, and that's what I'm doing in the last 2 terms of this equation. So the best fit model here, based on AIC, included only relatedness as an explanatory variable. It indicated that relatedness drives 21% of variability in associations between individuals. So there are likely other variables that could be included in this equation to improve our ability to predict associations. But what's important for our purposes today is that relatedness was found to be a significant driver of associations among individuals, which is not what you would expect in most natural populations. So to sum up this section, I found evidence for an insular main Hawaiian island population. And within that population, there are genetically distinct island communities. Now, I also found evidence for a reciprocal link between social and genetic structure. And that link likely acts to stabilize the current structure and accelerate local convergence. And then I found correlations between acoustic, social, and genetic structure. Okay, so we made it to the end. So I'm just going to wrap up by giving a little summary synthesis of everything that I presented today and talking about why it's important. So when you walk out of here, I want you to remember two things. The first is that Nysa and short-fin pilot whales are not only morphologically and genetically distinct, as we already knew. We now know that they have geographically distinct distributions throughout the Pacific Ocean. And those distributions might be separated by the desert of the North Pacific Gyre or by some sort of prey distribution. We know that they're acoustically distinct as well. Now, these two pieces of evidence support the hypothesis that Nysa and Shiho pilot whales are actually separate subspecies or maybe even species. The second thing I want you to remember is that I and my co-authors successfully applied the principles of gene culture coevolution theory to improve our understanding of population structure in a social whale. We found reciprocal links between genetic and social structure, and also found strong correlations between acoustic, genetic, and social structure. Now, this was the first time this method has ever been used to study short-fin pilot whales. And our success means that there's a lot of potential to use this method to study short-fin pilot whales throughout their range. And what's more, the results of this research support the application of gene culture coevolution theory to other social mammals, and indeed to social species across all taxa. And this is important because understanding the social or ecological mechanisms underlying a species population structure could improve our ability to manage and conserve those species. So, specifically for the case of Hawaiian pilot whales, these results suggest that in order to conserve the genetic diversity within this population, it may not be sufficient to conserve geographic populations. Instead, we may need to develop higher resolution monitoring based on their social structure. So thank you guys all very much for listening. A lot of people think. And so let's get to the fun part. So I've spent all of the years that I've been at Scripps thinking about all the things that I would say when it's time I was trying to get up here, thank all the people who got me here. But when I sat down to write this out, I started to get really overwhelmed by just how much there was to say and how little time there was to say that thing. So let's see how this goes. So I'll start with my advisors. Jay and Phil, I literally cannot put into words how grateful I am for your generosity over the years. Thank you first for sharing all of your professional expertise. But also, thank you for teaching me how to balance a scientific career with a healthy life. And thank you for teaching me to be ethical, both scientifically and professionally. And Jay, thank you for the woodworking lessons. If I ever need a backup career, I'm all set. Thank you both so, so very much. And Lisa, thank you so much for stepping up to co-advise me. I know that it takes a lot of flexibility to advise somebody when you have to give them such a long leash. But throughout these years, I've really appreciated all of your feedback and your support and your direction. And I've really enjoyed having a female role model on my committee. And then to the rest of my committee, to Ron and Bill and James, I turned to each of you for guidance at some point during my thesis. And I was always pleasantly surprised at how generous you were with your time and your expertise. I really appreciate all of your support. Thank you very, very much for taking the time. Okay, I owe a big debt of gratitude to all of my co-authors who have been wonderful to work with. especially Robin Baird. Literally, this project would not have been possible without Robin sharing all of his data and his time and his field project with me. Karen Martin and Marie Roche, it has been a great experience working with you both. Even though you weren't on my committee, you were both like mentors to me, and I thank you for it. And then to the marine mammal genetics group, you guys got me through some of the hardest days of my PhD. So thank you so much for training me to use such valuable data and for being patient with the inevitable lab mistakes that crop up. And the same is true for the acoustics lab, especially Shannon Rankin. Thank you so much for sharing all of your acoustical expertise with me. And to my fellow lab mates, Ms. Bayali, both past and present, thanks for sharing this wild ride with me. Erin, especially, thank you for being my partner in professional development all of these years. And to the grad office, to the grad office, thank you for being unwavering in your support for the grad students here. And a special thank you to Denise for being such a great friend and ally to TG, students, and to me. It means a lot. To CNBC, and especially to Penny Dockery, for giving me a chance to explore all of the integrative aspects of science. Thank you so much. And then there are some people at SIO who don't fall into any specific category, and yet are always generous with their guidance, starting with my scripts application and going all the way through my very last practice talk. Thank you to all of you for helping me navigate the winding road of grad school. And thank you to the many motivated interns and volunteers who dedicate their time to help me process data. Because without you guys, I'd probably still be in the lab. And then finally, thank you to all of the funders who came together to make this work possible. So now I have just a couple more slides, and they're in no particular order. But before I go through them, I wanted to give a shout out to one special group in my life. We came together in our second week at Scripps when our cohort lined up by the SIO pier to pick PO study groups. I know you guys remember that day. So our PO study group morphed into family dinners, and then family dinners became a jam band. And our little group grew larger and larger over the years, and through thick and thin, you have always been with me. So guys, this is for you. This is my favorite jam band song. I'm not saying that we sing it well. I'm saying that we sing it with a lot of love in our hearts. Okay. To my family, thank you, of course, for coming all the way to San Diego to celebrate this day with me. And thank you for sparking my curiosity about the natural world, a flame that will burn bright for the rest of my life. To the San Diego Mountain Rescue Team, you guys are more like a family than a team. Thank you for all of the lessons and adventures over the last 6 years. And to all of my adventure buddies, thank you guys for keeping me connected to the wild places. To Paul Dayton and everyone in our amazing natural history class, thank you for keeping the passion alive. Y'all are wonderful, and it was great to learn about the natural world through your curiosity. To Crystal, Julia, Peter, Kate, Megan, Spencer, Sean Crosby, Sean Haney, to Aaron and Mariella and Ray and Jess, and all of the amazing friends I've met at SAO. You guys bring joy to my life. So thank you for softball games, scavenger hunts, thank you for camping trips and backcountry adventures, family dinners, and game nights, every single holiday celebration. Thank you. So hard.
First of all, really great talk. And I just had one remaining question from all the questions that I've been throwing at you over the last week. In the Hawaii grouping of slides, you mentioned that you were only able to compare the Hawaii to Guam group and the Big Island to Maui group, or just Big Island group. And I was wondering why you couldn't use this as well.
Sure. That's actually just, at this point, a data limitation. So we know a lot about the social structure, but we still don't know everything. And in that area, there's some overlap between those two groups. So because there have been limited field projects in the kind of Oahu to Maui region, we don't have a lot of photo ID data or genetic samples from that area. So give it a couple more years and we'll get there.
I have a question about the nice and keto. So when we talk about the split between not maybe the gyre was some sort of barrier, what do you think explains the difference between the north and south off of Japan? And then did those splits happen at the same time, or did one see each other, or like, what did you think about this divergence?
So these are all really great questions, and maybe one day we'll be the topic of a future paper. But right now we don't actually know the answer to most of them. I speculate that the Nisa or the Shiho type group that you see in North Japan is just a small remnant population that found its way over there recently. They have their mitochondrial DNA is very, very similar to what we see in the Eastern Pacific, so they probably haven't been there long enough to diverge. But we don't know when they went over there, and we don't know why. Okay, who's next? Sarah.
So subspecies or species, and what would you need to do next?
Subspecies, I think. I feel like we've talked about this a lot throughout the center. They're different, but not as different. Luckily, at the Southwest Fisheries Science Center, they've been working for a long time on as you know, on a comprehensive guide to delineating the differences between species and subspecies. So using that, I think that we could pin it down pretty well. But next steps are to look at their full mitogenomes and nuclear structure across the entire globe, and that's actually something we're working on right now. We also, I think, would ideally collect additional acoustic and morphological data from regions throughout the Pacific and the rest of the world oceans to confirm that these links that we see between the different data types are found not only in the Pacific but also throughout the ocean. Barb.
So how do they deal with inbreeding? You know, I mean, this thing's set up to just like go down the toilet and make diversity. What's going on?
Yeah, it's incredible. I've always kind of worried about this myself, but I read a couple papers that suggested that while you think that social structure promotes inbreeding, it actually helps them. They have mechanisms they can use to avoid inbreeding. So they're just kind of selectively choosing who they mate with from a group of somewhat related but not too related individuals, if that makes sense. Okay, Eric.
Great talk, Amy.
Thanks.
I had a question about your mixture model.
Yeah.
Have you tried applying it to any data outside of your test and validation or your training test sets?
I did apply it to a couple different recordings that I had from the Central Pacific Ocean. And those weren't included in my training or test data originally because in that area of the Central Pacific, we are not sure whether there are Nisa or Shiho type individuals. So I tried to use those recordings to get an idea of what the distributional boundaries are there. Unfortunately, I can't ground truth them because I don't have any photos of those recordings, but I did find strong agreement using the bootstrap analysis. Yeah. Yes.
I heard you.
Yeah, go for it.
So great talk, thank you. This will probably
to show my ignorance of how whales are sampled, but I imagine that when you get your acoustic recordings, the whales that are not quite whales are within their social structure, so that should bias your sampling. Do you have any information of what happens when those whales encounter other groups? Do they become multilingual? Do they learn new languages? Do they adapt?
Yeah, that's an interesting question. So I had, I mentioned in my slide that I had 10 recordings from single species or single unit groups, and then 12 recordings from multi-unit groups. So I compared those to look for differences among them, and I found strong differentiation. And that's not enough yet to tell us exactly what's going on, but in addition to finding strong differentiation, I found some evidence for a lot kind of simpler call structure during the multi-unit encounters. So it's likely that, or I guess my speculative hypothesis based on that is that once they come together in multiple groups, they're using kind of repeated simple call types to maintain which group they belong to.
So you mentioned that both acoustics and genetics obviously change over time. So I was wondering what the range of time the functions were made for both acoustics and genetics, and then also if it's a huge range, you have to correct by time involved.
So we didn't correct for time, and this question is the subject of my up-and-coming book chapter. And the idea of quantitatively bringing together acoustic and genetic structure is something that people have kind of sought after for a long time, but not quite achieved. So what I would like to do in order to try to get at that is we can build phylogenetic trees, and we can build phyloacoustic trees, say, and then overlay those on top of each other. So if we group those in something like long-fin pilot whales, where we know how long ago they diverged, then we can use that to back calculate what the divergence rate is for acoustics, and then compare that to the genetic structure.
Thanks for your talk. This is kind of related to Jess's question. And between the different call types of communication versus like hunting type calls, is there a certain type of call that is more or less conserved between two species, two groups?
So I only looked at social calls. This is a great question. I don't know for sure. I would guess that it would be echolocation.
That's more conserved.
Yeah, but that's kind of a guess.
Well, I think we have to cut it off. There's a lot of events going on today, and you may have to run off to something else. But we're going to take a break now and adjourn, and we'll have a chance for your committee to ask you a few more questions, and then we'll all join together again for lunch at served time. Thank you.